This lecture we are going to have a look at the urinary system. So let me attempt to make some notes. So urinary system. And before you start listening to this lecture, make sure that you listen and have a look at the urinary system introduction PowerPoint, which is found on VLE, because if you do that, this lecture will make a lot more sense to you. So go and find urinary system introduction PowerPoint and have a look at that. So today we are looking at the urinary system in a bit more detail, and we're specifically looking at the structure of kidney in more detail. So here, instead of looking at the whole kidney, I'm going to kind of zoom into a single kidney and look at what it's made out of. And what it is made out of are millions and millions of tiny tubules called nephrons. So let me write it down. Nephron or nephrons. So as I said, you have literally millions of them per kidney. So millions per kidney. And you can also only see them under microscope. So they're a little bit of like a, I suppose, equivalent of alveoli in the lungs. So if you cut through a kidney, you won't be able to pick out individual nephron. So in order to be able to see them, you would need to have a really, really thin slice of kidney and then stick it under microscope, and then you'll be able to see cross sections through these nephrons. So there are two main processes which take place in the nephron. So let me write it down. So two processes that take place in nephrons. Right, I'm sorry if I misspell something, but I'm gonna have a go at not making many mistakes. And these two processes were actually briefly discussed in the introduction. So you should be able to recall these two names. So the first process is called ultrafiltration. So the bit ultra, that section, it simply refers to the fact that it's a filtration at a very, very, very tiny scale. So ultra is just very small scale filtration. So why are you filtering in the kidneys? Well, what you're doing is you are taking blood and you are filtering any small molecules out of your bloodstream, and those small molecules are going to end up inside a nephron. So what kind of small molecules could they be? So here you need to sort of really think about, well, can you name some of the small molecules which would be found in your bloodstream? So let's think about it together. So the first small molecule you could potentially name is water, right? So water. So if you think about the formula of water, this is H2O, and you remember that a molecule of water essentially contains three atoms, so two hydrogens, one oxygen, so three atoms altogether, and so that makes it a really small molecule, just three atoms. Now another small molecule, which is also small enough to get sort of through the gaps in that filtering system and get filtered out of your bloodstream, is glucose. Now glucose is a little bit bigger than water, but it's still a relatively small molecule. So if you think about again the formula for glucose, this will be C6H12O6. So here we've got 24 atoms, so a bit bigger, but again still small enough to be filtered out nevertheless. Right, another example of a molecule which is again small enough to get through this filtration system, is urea, right? So I'm not going to write down the formula for urea. That was discussed in that introductory PowerPoint. So again, it might be a good idea for you to look up your notes on that. And so, and then I'm just thinking about other small molecules. So another small molecule I could think of are the molecules which make up proteins. So you should be able to name those molecules. So the names of molecules which make up, which are like building blocks of proteins. So I'm really hopeful that quite a few of you will be able to name those. So these are called amino acids. Amino acids. And again, these tend to be acids, yeah, acids. So these tend to be pretty small, right? Maybe a little bit bigger than glucose, depends on which kind of amino acid you're talking about. But they're definitely, again, small enough to get through that filtration system. And then I'm going to throw in one more example. This is something called ions. Ions. Right, so ions. Again, you should remember from the beginning of the year what an ion is. So ion is an atom which either lost or gained one or more electrons. So here we are not talking about even a molecule, but we're talking about a single atom, right? So that's going to be smaller than urea and smaller than glucose and smaller than water. So we're talking about really, really tiny things. And they're going to be, again, definitely small enough to get through those gaps in the filtration system and end up inside the nephron and get filtered out of your bloodstream. So that's just the first step, right? So that's the first stage of a process. So we took all the small molecules, a majority of small molecules, out of our bloodstream and into the nephron. But that's not enough. Here you have to have the second stage of a process, and this stage is known as selective reabsorption. So let me write it down while my clock is doing its thing of cuckooing. So selective reabsorption. Let me just erase that. It's gonna be funny. Selective reabsorption. Reabsorption. Right. So here what I'm doing is I'm then taking some of the useful molecules which I filtered out and putting them back into the bloodstream, because some of these molecules I want to keep and some of them I want to get rid of. And so here is where that selective bit comes in. So you're sort of selecting which ones you're going to be keeping in your body and reabsorbing back into your bloodstream, and which ones you are going to keep inside the nephron, and therefore you're going to weave them out as a part of your urine. Okay, so here we need to kind of have a bit of a discussion about which molecules we are going to keep and which ones we're going to get rid of. Well, let's start with the first one, which is glucose. So what do you think? Do you think we want to wee that glucose out and lose it, or do we want to keep it in our body and reabsorb it back into the bloodstream? Well, the answer, I hope, should be fairly obvious to you. I definitely want to keep all of the glucose. Why is that? Because I'm using glucose for the process called aerobic respiration to produce energy. So I've kind of spent all this effort to digest carbohydrates, to break them up into glucose, and I then took this glucose into my bloodstream, and I'm going to be using it to produce energy, to power my brain, to power my muscles, to power all of my organs. So I definitely don't want to get rid of any. So I want to reabsorb it all. When it comes to amino acids, well, they are building blocks of your proteins. So again, useful little molecules. So I will want to keep them. Then let's talk about some of the other molecules. What about urea? Well, to make decision about urea, you again need to go back to that introduction where we talked about what urea is and where it comes from. So I'm going to leave that to you to kind of look up if you can't quite remember. But you will see that urea essentially is a toxic product. So this is not something you want to have in your body in large quantities. So this is something I do want to keep inside the nephron and therefore not reabsorb, and that urea is going to be then become part of my urine, right? And so this is, you see, where the selective bit comes in, right? So I'm sort of selecting which bits to reabsorb back into the bloodstream and which bit to wee out. So let's again talk about other things. So what about ions? Well, ions, I do need some, some of the ions, definitely, because I can use ions in processes such as muscular contraction, in processes such as nervous impulse conduction. So I definitely need some ions, but I might not need as much as I have in my body at that point in time. Maybe I ate particular diet and I have too much of one particular kind of iron and I don't need it, or more than one particular kind of iron. So your body, your kidneys are going to kind of decide, in inverted commas, how much of each type of iron to keep, because you might need that for that particular, at this particular point in time, for these different processes, and how much of these ions are in excess and therefore you can afford to lose them. And the same kind of logic, I hope you will be able to figure it out, that the same logic applies to water. So if I drank recently and have too much water in my bloodstream, I will want to retain some of that inside the nephron and not necessarily reabsorb that water. And then if you drank a while ago, you might want to reabsorb pretty much all of it, or higher proportion of that water. So it just depends on the situation of what you're doing at that particular point in time and how much water is found in your bloodstream. So I hope that kind of stage is also fairly clear. So what I'm going to do in this video is I'm going to look at ultrafiltration in more detail, and then in the next video I'm going to look at the second process, which is selective reabsorption. So let me sort of scroll down a bit, because what I'm going to start doing here is I'm going to start drawing the first section of a nephron. Now, nephrons are very complex structures, they're pretty long, but ultrafiltration, that first step, takes place in the very beginning of a nephron, and that's what I'm going to attempt to draw here. So make sure you leave yourself quite a bit of space around for labels and things like that, and hope I'm not going to make too much of a mess with it. So what I'm going to have a go drawing here is essentially something which looks a little bit like a top of a chicken bone, and this is a cross section of that first bit of a nephron, and the whole thing looks a little bit like essentially a cauldron, because that's what filtration is, isn't it? You're sort of putting some stuff into a cauldron, and then smaller particles are gonna kind of get past that cauldron and out. So that's what that first section of a nephron is. So just to give you a little bit of sort of visual, so we're all on the same page, and I know some of them are really good at visualizing things, and maybe some of you are a little bit worse at it. So let's have a look at it together, right? So very simple picture. So here I've cooked some pasta and I've put it into a cauldron, and that's essentially what that first section of my nephron is going to be, right? So imagine if I do a cross section of that cauldron, that's the kind of shape I'm going to end up with, right? And here you can clearly see that smaller things like droplets of water, maybe a little bit of starch, maybe tiny bits of broken off pasta, anything basically which is small enough to get through these holes is going to get drained, and then the bigger stuff, the actual pasta, is going to stay inside the cauldron, so that's not going anywhere. Now, this is easy enough to kind of relate to and visualize, but I would like to ask you one question. So when it comes to this picture, to this pasta getting drained, what is the force which is causing that liquid, that water with a bit of starch, what is causing it to drop down into the sink, right? And I'm hoping that quite a few of you are going to be shouting at the video saying, I know what it is. This force is gravity. And if that's what you're thinking, then you're absolutely right. So here I've got gravity causing those smaller particles, right, those droplets of water and tiny bits of pasta to drain out of the cauldron and away from the stuff I want to keep, right, from this pasta. Now, that force is not going to be the one which I could apply in the nephron. Well, why is that? Well, because if you think about shape of a kidney, right, so some of the nephrons might be the right way up, where gravity could have potentially acted on that filtering system and forced smaller particles through. But you also have some nephrons which would be, well, essentially upside down, right? So they would be working against gravity. So that's not going to be, gravity is not going to be the answer. It's not going to be the force you can feasibly use to get the smaller particles through. So you need to have some kind of other force which is acting on the nephron, right? So let me just go back to my drawing. So this is my kind of cauldron, if you like, right? So that's beginning of that nephron, the beginning of that filtering system. So that first kind of cauldron-like section, it's got name to it. So this one is called Bowman's capsule. So let me write it down. So Bowman, that's an apostrophe, by the way. I've done my best at drawing it. There you go. Bowman's capsule. So when it comes to spelling, by the way, I plenty of time I put comments in the assignment saying, please do not spell biological terminology with capitals. A lot of people end up spelling carbon dioxide or I do not know, esophagus with a capital, and I'm saying no, 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 it's all lowercase letters. Well, here Bauman's the B is actually capital, and I hope you know why, because it's named after a person, hence the apostrophe. So it's a capsule belonging to this chap called Bauman, because he was the scientist who kind of first described it, and so that structure ended up being named after him. And then further on, yes, when you leave that cauldron, you're going to arrive to the first section of nephron beyond Bowman's capsule. And this first section is called proximal convoluted tubule. I know, it's a bit of a mouthful, isn't it? So proximal convoluted tubule, right? So imagine if you're a doctor who deals with urinary system issues, right? You don't want to be the one who is saying proximal convoluted tubule, a few times a day. So because of that, this structure is actually abbreviated to PCT. Okay, so PCT is a common abbreviation which you might come across in books, etc., etc. So what's happening here in this kind of dip of a Bowman's capsule? So what you have here is a system of kind of blood supply, because that's the blood which is going to be arriving into the nephron to be filtered. So I'm going to use red color here just because we associate blood with red color, so it's just easier for it to stand out a bit. So I'm going to show you the first blood vessel which is going to be bringing blood into that top bit of Bowman's capsule. And this blood vessel is called afferent arteriole. Oh, there you go. Afferent arteriole. So blood is going to be coming in along this afferent arteriole and into that deep or imagine like that cauldron shape of Bowman's capsule. So here, that afferent arteriole is going to split into smaller blood vessels, which are capillaries. So I'm going to just sort of show you like a triangle of these capillaries. Okay, so it's going to be plenty of these capillaries here. And then those capillaries are going to get gathered back into slightly bigger blood vessel, which is another arteriole. And this arteriole is going to be taking blood away from the system, and this one is known as efferent arteriole. Efferent arteriole. There you go. Right. So afferent bringing its blood in, efferent is taking it out. So as we discussed before, so any yes, I think I'm going to leave it that kind of size. Any smaller molecules which were inside those capillaries, they are going to come out of a capillary and they're going to end up here inside Bowman's capsule, and they're going to start traveling down into proximal convoluted tubule. So that's where you're going to find things like water, things like glucose, H2O6, things like amino acids, things like ions, and things like urea, right? Urea. So that's going to be the kind of a mixture of some of these molecules. And these molecules collectively, this sort of soup of smaller molecules, it's known as glomerular filtrate. So let me have a go at writing that. So slightly smaller size. Hope that works. So it's called glomerular filtrate. Also sometimes known as ultrafiltrate. You remember we're talking about ultrafiltration, so you might come across this alternative term as well. So let me go back to black color. So let's think together. You remember my picture of a cauldron, right? And the fact that we were discussing that the force which was causing that water to drip out of the cauldron and through the holes was gravity, right? But we also discussed that gravity is not going to be the answer when it comes to a nephron. So I need something else, some kind of other force or forces which are going to be causing those smaller molecules to get filtered out. Because if I didn't have any force at all, then those molecules are just going to keep on traveling through that system, and they're never going to get filtered. So I need something here, a bit of a kind of push somehow, which would cause those smaller molecules to get out. And if you look really carefully at my drawing, it could actually give you an answer to what that force might be. So again, I'd like you to kind of pause the video at this point and have a bit of a think, right? So think about what is on that diagram. Can you see something which is going to be causing these smaller molecules to get through that system into Bowman's capsule and become glomerular filtrate? Okay, and I hope those of you with powers of Sherlock Holmes are able to see that there is a bit of a difference, quite a marked difference, between the sizes of my two arterioles. Yeah, how many of you noticed that? Can you see my afferent arteriole is bigger in diameter compared to my efferent arteriole? And you can imagine what's going to be happening. So you have bigger tube, essentially, bringing blood into the system, but then smaller tube, which is allowing some of this liquid, or blood in this case, to leave. So what you're going to have happening is that there's going to be a build-up of pressure, because some of these molecules essentially have nowhere to go other than to be forced out and into the Bowman's capsule. So this is one force which is causing those molecules to get filtered out. And obviously then you also have a second force, because if that liquid was just stationary and if it wasn't kind of forced into the system in the first place, well then nothing would be filtering out either. So you need to have this initial force for the movement of blood. And if you think about what is forcing blood through your blood vessels, well, essentially it's your heart, right? And the action of muscular walls of arteries and arterioles. So that's going to be another kind of part of that puzzle, right? So another force which is causing those molecules to get filtered out. So the next question is, well, okay, so we understand what is causing these molecules to get filtered, but what is the actual system of filters here? So you remember in the cauldron, so let me go back just for a second to this picture of a cauldron. So here, what I have is as a barrier, right, which is the actual cauldron, and in this barrier I have circular holes, and they're of a particular size, and this is what's allowing that water, starchy water, to drain out. So I need something similar. So I need some kind of membrane, some kind of barrier which is standing in the way of bigger stuff getting through. And at the same time, I need holes of certain size which would allow things, as I said, glucose and water to get through. So what is my system of barriers in a nephron? Okay. So in your nephron, you have 3 barriers of filtration. So let me write it down. So 3 barriers of filtration. Filtration. Okay, so 3 barriers of filtration. There you go. Got there in the end. So before you go any further, can you tell me what 2 out of these 3 barriers of filtration are? And I'm not asking for any kind of technical answer. All I'm asking is, okay, let's have a look at this diagram really carefully and think together. So if I have something inside that blood vessel, what is in the way which is stopping everything getting inside the Bowman's capsule? So what do you notice on that diagram which is sort of stands in the way of that blood inside my blood vessel? What is stopping it from ending up here? Okay? So you should be able to, by just looking at this picture and think about it logically, give me the names of 2 barriers of filtration, right? No more, no less than that. So again, I'd like you to kind of pause the video and have a little think about it. And I'm really hopeful that you will be able to arrive with the answer. I'm not asking anything, any new terminology or something super complicated. You don't have to start Googling it or anything like that. You just literally think about it in a logical way. Okay, so as I said, let's sort of think about it, right? So if I really zoom into my picture, so imagine I've got some blood inside that tiny blood vessel, which is one of my capillaries, right? So in order for this stuff to get from inside that capillary into this space, what do I have to get through? Well, the first barrier will be the wall of the actual capillary there, right? Because it's such a thin line, right? You have to imagine that that's just a blood vessel. But that blood vessel has got walls, right? And those walls are going to be stopping any big stuff from getting through. So that's the first barrier. And then the second barrier is just that black line, isn't it? That's the kind of the wall, if you like, of Bowman's capsule itself. So that gives you 2 barriers of filtration. So let me just zoom out again. By the way, I also need to name one more structure on this diagram. So let me do that before I forget. So I'm going to go back to red color for a second. Let me check it's the right size. Yeah, it's small. Okay, so that triangle of capillaries, which I've drawn earlier on, it also has got a name to it. So I'm going to do another label line coming from that triangle of capillaries. And that triangle of capillaries, so-called capillary bed, that's how it's sometimes described in books. But it's got this special name called glomerulus. Glomerulus. And hence, you remember that term I mentioned earlier, glomerular filtrate. So the glomerular filtrate is essentially named after that capillary bed, glomerulus. Okay, so going back to three barriers of filtration again. So this time, because I've sort of run out of space here somewhat, I'm going to scroll that way and then start numbering my barriers of filtration. So my first barrier of filtration was capillary wall. Capillary wall. And this is capillary, which is found inside the glomerulus. So I'll put it in brackets. So glomerulus, right? And here I'm literally just giving you one bullet point, just 3 words. But your job is to do a bit of research, so maybe find out about how how big the molecules can be to get through those holes, what they're called, I don't know, just give me something, 1 or 2 or 3 sentences to describe that first barrier of filtration. So the second barrier of filtration, as we sort of described and pointed out earlier, is that black line, essentially like a wall of Bowman's capsule. This is referred to as basement membrane of Bowman's capsule. Okay. But there is a third barrier of filtration which I cannot draw on my diagram because it's pretty tricky to draw, and I'll show you why. And but that barrier filtration is known as specialized cells called podocytes. Podocytes. So what podocytes are, they are very kind of specifically shaped cells which are found surrounding those capillaries of glomerulus. And to show them to you, I'll have to give you example of a drawing of a podocyte. So give me a minute here. So I'm going to go on to the next slide. So here I've got a picture of a podocyte. So imagine that's the capillary and the glomerulus. And so what podocyte is like this really peculiarly shaped cell which is sort of wraps around the capillary. I sometimes give people this analogy. I think it looks a little bit like a crocodile clip, which you clip your hair with, where you've got teeth kind of interlinking with each other. And that's what podocyte is doing, sort of wrapped around this capillary. And it's there's got these extensions which are sort of interlinking. And what you end up with are lots of really, really tiny gaps between these sort of, in inverted commas, teeth. And so any molecules which are small enough to get through those tiny gaps, they're going to be the ones which are going to end up inside the Bowman's capsule, and they're the ones which are going to carry on traveling through proximal convoluted tubule, etc. So where you've got lots of podocytes together, this is sort of what the whole thing would look like. But if you take a single podocyte, that's what it would look like as well. So here I've got a micrograph actually of what these sort of structures of podocytes look like in real life. So this is electronic micrograph of podocytes and Bowman's capsule. So that gives you kind of the third barrier of filtration. Okay, so let me go back to this list. So essentially what you have then is not just kind of one cauldron, but essentially like a series of three stages of filtration. So you're really, really, really making sure that only the right molecules are going to end up inside Bowman's capsule. So let's therefore think about it together. So what about things like, let's say, big proteins? So something like, I do not know, or let me give you another example, or red blood cells. That's one of the common questions I ask students. So let's take red blood cells. Would you expect red blood cells to penetrate through this series of filters and end up inside the proximal convoluted tubule? And the answer is no, right? Because those molecules we were talking about, they're really tiny, whereas if you think about molecule of hemoglobin, just a single molecule of hemoglobin is much, much bigger than size of these molecules, something like water or glucose. And then a single red blood cell, you remember, that contains millions of individual hemoglobin molecules. So again, this is not something which you would be able to get through these barriers of filtration. So therefore, if you have some blood in your urine, right, or your patient has got blood in their urine, that means that there is a problem, right? So for some reason that system is not working. For some reason there is some kind of breach in that series of filters, and so the blood is able to kind of get into the nephrons and therefore end up in the urine, which should not be happening in a healthy kidney. So that could be a sign of infection or sign some kind of underlying issue. Okay, so let me just then give you some numbers. So when it comes to filtration, so how much blood gets filtered in your kidneys? Well, in 1 minute, so this is per minute, so in 1 minute. In 1 minute, you have the following quantities getting filtered. And this is per both kidneys, right? So we're not talking about one kidney, but both, the whole of your urinary system. So per 1 minute, you have 200 milliliters of blood arriving into your kidneys to be filtered. So this is how much blood gets serviced, if you like, by your kidney every single minute. So how much of this blood is going to end up, getting filtered out and become glomerular filtrate, right? Well, out of those 200 milliliters, round about 125 milliliters is going to turn into glomerular filtrate. So glomerular filtrate. Right? So the rest of it is just going to stay in the glomerulus and leave afferent arteriole. Well, imagine now if all of this filtrate became urine, because you remember anything which ends up inside the nephron can potentially become urine. So, that's obviously is not going to be sustainable, right? If that was reality, all you would be doing is just sitting on the toilet and constantly drinking water and constantly weeing, right? And your whole life would be just dedicated to that, which is luckily is not the case. So you can see that absolute majority of that filtrate is going to be reabsorbed back into your bloodstream. So only round about 1 milliliter of that glomerular filtrate is going to turn into urine. Obviously, these are average numbers, right? So it will depend what specifically you're doing at that point in time. If you're exercising and losing some more water through your sweat, it might be small amount. If you drank a lot recently, it might be big amount. But we're talking about average numbers just to give you a flavor. So therefore, if you look carefully at the numbers, right, how much of this filtrate are you going to be reabsorbing back into your bloodstream? So very simple maths. I hope you can figure it out. So therefore, you're going to end up with round about 124 milliliters of filtrate getting reabsorbed every minute. So can you see, can you just appreciate just how much work your kidneys are doing for you every single minute of your life, from your birth all the way to to the time you're really old. And so it's just tremendous amount of work. And you can also probably appreciate that if somebody has got kidney failure and their kidneys are not able to do that work, it's going to be just an absolute disaster for your body. So your body is just going to start holding on to all this toxin, holding on to all this excess water, and it's just not going to be a good outcome. So just to give you a little bit of kind of visual of what these numbers look like, I mean, one good thing you could do is actually, if you have a measuring jug, is actually measure those amounts of in water, and just to see, to show yourself, what is the relatives amount we're talking about. But just to give you a little flavor of what these numbers look like in terms of quantities. So I've got this picture which I found of 3 different sized measuring cylinders, and just for kind of scale purposes, so you can see somebody's holding one of these bigger measuring cylinders in their hand. So this is what they would look like. So that measuring cylinder, around by that amount, would be 200 milliliters, right? So you can see it there. So if you take smaller cylinder, right, so that would be 125 milliliters of your glomerular filtrate, and you can see that that's going to turn into a single milliliter. So this really tiny little cylinder, and right at the bottom, that cylinder holds 10 milliliters, and so that just a little bit at the bottom of that cylinder is going to be 1 milliliter of resulting urine. So that just gives you sort of an idea of the type of work which your urinary system has to perform. So in the next lecture, what we're going to do is we're going to look at the selective reabsorption. So you remember that we started by saying you've got 2 processes, ultrafiltration and selective reabsorption. So we've now done that first bit, but that leaves us with this process of selective reabsorption to discuss next.